That's every, yeah, so that one. Just now they all use the Presto card.
Benchmark with our assignment.
Yeah.
You just ignore the zero. I don't know what's going on. I said it's not consistent result.
You've been able to run tests, but they just don't make sense.
Every time I run a test, it gives me a different result. It's not the same as the test.
Yeah, it's about polar bears or something like that.
Yeah. It's 3 times difference.
I don't know what they're doing for their OS.
Something is OPA. I try single GPU. It's
Oh,
okay.
but the OPA, but anything off node is worse, or is it
So much
worse, much worse?
much worse now. I don't know what they do when they're down.
Well, I know in their last upgrade, they went, there was a bunch of OPA, they went to 7.5 CentOS or something. I think the OS upgraded, and so they had to upgrade all the OPA drivers, and there was some issues. So maybe they haven't resolved it, because they were having a problem where if one of the OPA nodes went down, it took the whole switch. Like, so there's like some sort of, it sounds like a fabric manager problem. So I know they were, that was, when did they had that shut down, what, about a month ago? So maybe they're still working on it. But the single node, single GPU, like the 2 and 4, they look similar to what you get on the brand. Yeah, because it should be about the same, right? The same sort of hardware, same processor, same GPUs.
A larger memory one.
But in terms of apples to apples, right? I mean, it should be. The only difference is Dell versus a Huawei. It's the same machine, right? Same box, and same.
Yeah, I didn't really know it's there. Something with the switch, QPA, QPR interface. That's not hardware issue. It's the software.
Sorry, the software.
Do you want me to get started? Or?
No, well, let's just wait. Check out Ramsey's probably devastating. All right. So we're back. It's working.
want to introduce, or shall I?
Go ahead.
Sorry, so welcome everybody to the Sign Out User Group meeting. Today we're having a speaker. I'm very privileged to introduce. She was actually our student last semester at Scarborough, Stephanie Pank. She, your DPS?
EEB.
EEB, Environmental
Ecology and Evolutionary
Thank you.
Biology.
Yes. something. She studies polar bear weight models. Well, you're going to tell us about that. So anyway, so she's a PhD candidate, and we're very privileged to have her here today, since not very often we get one of our students up here to give us a talk. So Stephanie, please take it away.
Let's see here. Maybe I might be able to take it away.
How do we make it big? If you go to view, can you make it that way? Who uses PowerPoint?
It should just be this button down here, no?
That's usually all you do.
Yeah, right, okay, I'm not crazy.
Yeah, that's the way to view.
Let's see here. It's computer.
Yeah, we feel the same way.
Slideshow.
Oh, yeah.
Where's slideshow?
Up at the top.
Oh,
Oh, slideshow.
No. Oh,
From beginning, yeah.
from the beginning. As well, though, you could run.
I don't know.
Is something stopping it from maximizing it? 'Cause you're a
Yeah.
viewer?
There are two instances of PowerPoint. Maybe it's
Under two there? What's going on there?
Oh, yeah, that's right.
Click on that. Yeah, just click on that.
Yeah,
Yeah.
Oh, it's probably on screen too.
I think it's because it's on screen too. We've got it split.
Why do you have a screen too?
I don't know. It shouldn't be.
People.
so I just,
Just a second. It's just it's
Yeah,
off.
it's just yeah.
Close it, yeah, you can close that.
It's just because of the way the two screens are not splitting the image properly.
it should be, let's see.
It's not you. It's us.
It's thinking it's on a screen, and so it's why it's.
That's right, I'm starting by telling you guys how I'm a biologist and not good with computers, so. It's good preface.
We'll say it's a Windows system. We wash our hands of it.
Oh.
Try it.
Okay. Success.
Yay.
Okay. So, hi, I'm Stephanie. As Eric mentioned, I started my PhD 2.5 years ago now. And I am primarily a biologist. I only recently started looking into mathematical modeling and actually using them in this PhD. I did not have a background in it, so I think that's a major point about this Making a Modeler, aside from the fact that I was watching Making a Murderer when I created this talk, so it inspired me. But yeah, I did not start out knowing anything about models, minimal math background and minimal computer science background. So I still managed to make use of the Niagara supercomputer here, and I just kind of want to tell you a little bit about that story. So I did my undergraduate degree at the University of Guelph in wildlife biology. And then I graduated and went to work as a bat biologist. Yes, that job exists. I went out and caught bats for endangered species surveys. This is a flying squirrel, not the target species, but they often get caught in the nets. And basically what had happened is that my graduation from Guelph had coincided with an epidemic in the bat populations in North America. So lots of jobs catching bats, not good for the bats. But what was happening is that in around 2006, they think, cavers brought over a fungus from Europe. It's called Whitenose fungus. And basically this was a novel parasite on the bats. And they just started dropping like flies, basically. People would go into caves following a winter torpor bouts, and over 90% of some colonies had mortality. So this caused, obviously, a bit of a panic. Some colonies were completely wiped out. And the reason that people cared, aside from the intrinsic value of wildlife, is that it was rapidly spreading. So you can kind of see the epicenter was around New York, and it just started spreading out every year. And it's still progressing westward and northward. And again, the reason that people really cared is that bats are insectivores, at least in North America. All of our bat species eat insects. They're the #1 nighttime pest controller. So it kind of started a lot of panic in agriculture, in the agriculture arena, because they're worried about the economic cost of losing this natural pest controller. So I kind of started being on the forefront of this change, drastic change in the ecosystem. Started wondering about just how the loss of bats would affect the whole ecosystem. We know they eat insects, but exactly what insects are they eating, and how does that impact the whole system? So they eat mites, which are herbivores. They also eat some spiders, that's the harvestman spider, moths, katydids, and beetles. But when you look at these insects, you have to consider that not all of them are actually eating plants. So these 3 insects actually are known to eat other insects, which typically are the smaller herbivorous insects. So if you no longer have bats eating these mesopredators, there could potentially be some release, and maybe they could start controlling the herbivorous insects. And again, we don't know what the bat's predator, what are owls going to do? What are the species going to do that were using bats as their primary food source? Maybe they're going to prey switch and start controlling some of these pests that historically the bats were eating. So watching this whole drama unfold gave me kind of a front seat to the difficulties that ecologists face when trying to untangle and predict ecological phenomena. So there are some experiments that we can do. So there are controlled experiments. For example, some of the panic about the loss of bats started from some exclusion netting experiments. So basically what they do is go into a experimental area and exclude bats. So just put netting around a certain patch of the area that prevented bats from being there, and then basically look at how those 2 plots would differ, the ones without netting, the ones with netting. And that was some of the experiments that actually led to finding that there's an increase in spiders, katydids, and these other insects in there. But that's short term. And then we can also use model systems. So we really like to use Daphnia because you can have short generation times. You can explore a lot of theory with them. But again, this is a model species, and you can't do this with a lot of animals from the ecosystems that we work with. So there's also some natural experiments. We tend to not want to interfere with ecosystems anymore like we used to, but there's still certain things that we can observe. So in Yellowstone, we have information on what that ecosystem was like while there were no wolves, and then following the reintroduction of wolves. So this is kind of a natural experiment that we can look at before and after information. We also have a lot of invasive species occurring right now. So this is again a natural experiment where you can look at what that ecosystem was doing before an invasive plant came in and what's happening after, or invasive animal, be it whatever it may. And so although we can gain a lot of information from this, there are some problems. So for example, basically you can end up with only seeing part. Let's see here. We might only see part of the whole story. So again, kind of going back to these bats, we know that there's more insects, but we don't really know what's going to happen past that, or what the owls are going to do, or what the other insects are going to do. And it's also short scale, so you might miss these natural ecosystem cyclings, like wildfires can be a huge component of an ecosystem function. And when you're only looking over that short time scale, you can't actually get the full picture. So aside from experiments, we also just have long-term data sets that we can look at. We have a lot of data, abiotic data, so just climate data, which is very nice. We can also look at population level data, a lot of systems. We have the total number in a population, birth rates or mortality rates, from which we can get a lot of information. And we also have individual level data. So we can get age of individuals, health index, size measurements, weights, and individual level reproduction. So there are a lot of data sources, but it's just hard to put these things together. So we learn a lot from the isolated data, but then we're left with this struggle of how do they all fit together and what do they mean as a total, not just the parts. So we might be missing key links from the ecosystem, or if we have a complete picture of that model system, we don't know if that applies broadly. Does the Daphnia system react the same way when we look at the bats? Probably not, but how can we use this information to make conclusions? No matter what, we have a lot of uncertainty. So I started looking at this bat problem and basically came across ecological modeling processes. And this is the first time that I really started to look at modeling, and this is like maybe 3.5 years ago. So basically there is a whole slew of different modeling approaches going on in ecology. From on the left, we see dynamic energy budget model, and they're actually looking at individual level energetics and creating predictive models to understand biological processes like reproduction or growth, how much those cost. And then on the other side of the spectrum, we have predictive modeling, a little plug for Mars there, but actually like whole scale ecosystem where we're using abiotic data to predict phytoplankton blooms and actually do future scenarios after having fit that model with existing data. So what we can do is we have different ideas and we can do model simulations using the programming tools that we have at our fingertips, and kind of compare how these different hypotheses fit with the data that we have available and see if we can tease apart more meaning from those puzzle pieces. So I thought that this was really cool and something that might be useful with the white-nosed bat problem. So I got excited, made a grant proposal, applied, and I got accepted, but to look at polar bears. So bats, bears, both B, probably same. But basically I applied and my supervisor liked the idea, thought I could maybe start to understand ecological modeling, but he didn't know anything about bats, but he said I have bears. So I took on a project that's looking at the energetics in polar bears. But the point I really want you to take home from that little story is that I really had no modeling experience and minimal coding experience. I'd done a little bit of R coding in my undergrad. So I had some basic math knowledge and really an ecologist that thought modeling could help me understand things better going forward. So my system now. So the Arctic ecosystem. I think I've probably convinced you that ecosystems are complicated, and the Arctic is considered relatively simple. Even then, there's dozens of connections, and the connections themselves have many variables that impact how they work. And here we even have sea ice that is naturally variable, both over space and time. And that is a crucial component as it provides a platform for a lot of the Arctic life. And it's dictated by abiotic factors. So simple system, sure, but we still have a lot going on that we need to understand. Climate change is another factor here. It affects the sea ice extent and concentration, which indicates how useful the ice can be as a platform. So on the left, we have the sea ice concentration in February 2016. And these little red lines just show the median ice edge from 1980 to 2010. So you'll notice that it actually doesn't reach the average. And that is when the sea ice is at its peak. And then on the right, we have the sea ice concentration in September 2016, and that's when it's at its minimum in the Arctic. So it's a big change, and polar bears have to deal with this drastic shift in available habitat every year, regardless of what climate change is doing to that shift. So that is seasonal sea ice, the ice that disappears yearly. And polar bears living in these areas have to deal with it. So the Western Hudson Bay population is really on the forefront of seasonal sea ice. You can see here, that's their habitat area, and the ice completely disappears. And they're forced ashore for 4 months on average now, but that's changing, again, depends on when the ice comes back. So basically when they're on ice, they have access to, no, that's going backwards, was it? Sorry, guys. Basically, when they're on ice, yeah, that shouldn't have happened. Sorry, it went backwards, and I'm kind of confused. There we go. Weird. Okay, so when they're on ice, they have access to their main food source, which is really high-fat seals. And they're able to build up a lot of fat stores at this time. This is kind of their surplus season. And then when they're forced ashore, they don't have a reliable food source. There's some berries, lichens, and maybe they come across a whale carcass, and that's the jackpot. But they have to be able to survive off the fat stores that they make in the surplus when they're in the fasting season. So sea ice loss directly impacts the bear's ability to successfully obtain enough energy during that surplus season, and that affects their ability to survive and to reproduce. So the timing of the seasonal sea ice freeze up and breakup periods dictate the length of that onshore fasting season, and thus determines how successful they are. And those freeze up times and breakup times are dictated by temperature. So, add into the fact that the Arctic is warming at twice the global rate, we're already seeing a decline in the sea ice quality, abundance, type, and distribution. Here we see projections for the Arctic sea ice extent. And even the most optimistic projections show a large decrease by 2100. And then in red, we see the actual observations plotted on there. And the observations are actually worse after 2000 than the worst model projections. So it translates to a massive loss of that sea ice habitat that polar bears depend on for proper functioning of their biological processes. So this is a huge change in the abiotic components of this already complicated Arctic system. So I'm trying to use mathematical modeling approaches to ask questions about the polar bears that are having to deal with this ecosystem. So the questions I'm going to talk to you about today are, first of all, parasites of polar bears. So Trichinella nativa is a parasitic worm that are found across all polar bear species, and I want to know how it's maintained in polar bears. Exactly what's going on there. Is it going to be more of a problem under climate change? What's the story? And the second question is, how do polar bears assimilate and use energy for biological processes? So I touched briefly on those energy budget models. That's what I'm trying to do with the polar bears. Okay, so our first question. So Trichinella nativa is a nematode parasitic worm. It's appeared in the literature since the 20th century. Explorers were getting trichinosis from seemingly healthy bears and walruses and then dying horrible deaths. So it shows up quite a bit. And that led to a big investigation into exactly how that parasite life cycle works. And what they found is that when you ingest infected meat, you have there's larvae in that meat. When it hits your stomach acid, is anyone still eating? When it hits your stomach acid, it activates and actually turns into the adult worm, and you get a breeding pair of adult worms in your intestine, which then produce more larvae, and those larvae migrate to striated muscle tissue, and there they insist, and they're in that host for life now until something consumes that flesh, and then the process is started over again. So what we do know is how it happens, but not where these polar bears are getting it from. So that's just the cyst. And then, yeah, there's lots of uncertainty in this sylvatic cycle, the natural cycle. What's maintaining these worms? If it's just eating, who's the source?
Do polar bears die from it or not?
No. So they actually are able, and that's why the question kind of stopped there, because the polar bears, they seem to have no effects. Although I do have my doubts, because some of these bears have 9 larvae per gram tissue in some of their muscles. I find it hard to believe that that's not going to impair your ability to move. But yeah, we'll see. So we were interested in where it was coming from. So all of the polar bear populations that have been tested for T. nativa have been found to have high prevalence. And prevalence is simply the number of infected bears in the total, compared to the total population. So if we can figure out where these bears who seem to have a ton of it are getting it from, we could probably better understand how that sylvatic cycle is functioning. So to answer this question, my lab and I combined mathematical modeling framework with data that was already readily available from the literature. And so the 4 major hypotheses that come up when you look at T. nativa is that it's coming from their seal prey, or it's coming from consuming carnivorous carrion, which can include unsuspecting animals like voles or rabbits who take a meat meal. And both of these sources are interspecific, and so we considered them together in one model framework, Model A. And then there's also, so these hypotheses seem to have some traction in the early literature. And then when we move on to about 1960, it changes, the tone in the literature changes, and people are really into intraspecific sources like eating dead polar bears, so conspecific scavenging, and also cannibalizing. So they're basically saying the polar bears are giving it to the polar bears, and that's how it's maintained. So we consider the intraspecific mechanisms in a second model framework. And then we also add in age structure in the model, because there does seem to be a buildup over the age classes. So maybe it's just more time, more odds you're going to come across it, and that's the answer. So the major difference in these 4 modes of transmission just lies in the amount of contact a bear has with these different food sources, and with the different infected food sources. So our candidate models become, we have interspecific model A, which is simply a rate of contact with infected seals plus the rate of contact with dead carnivorous carrion. And then we have our intraspecific model B, which is just consuming conspecific dead and cannibalizing infected bears. And these were a play on an SIR model. So the output that we get from each model is a prevalence. And since we have prevalence from the literature, what we can do is compare the observed prevalence in the literature to the prevalence that's been simulated by the different mechanisms in our models. And you'll notice if you set seals to 0, you can get the prevalence that's only created by carrion. So we can still examine the 4 different ones as well. So why did I need to use SciNet for this? It's 2 models. What's the big deal? Just run them. Well, there's a lot of uncertainty because we weren't able to go out and collect data. We were really cobbling together a lot of our parameter estimates from the literature and from expert opinion. So we had a lot of assumptions, and what we needed to do is explore a ton of parameter space to make sure that any conclusions we made were robust across that large space. Okay. And we also wanted to know, when we came up with a model that we liked, what parameters were was the model sensitive to, because this can help direct future research instead of, if there's a parameter that we changed 100-fold and it didn't create any difference, then we don't need to look at that parameter for future research. And so I could actually do that fine with Model A, but Model B had really long running times. And so what we needed to do, since we had to run it hundreds of times, it was best to do that on SciNet, where we could send up hundreds of jobs at the same time. And also, any redos. We found a couple problems in our model structure and had to redo all the analysis. And that is painful if you've spent hours doing the runs individually on your own laptop. So it was worth putting in a little bit of extra effort to set it up for the SciNet computer. So how did we do that? Well, it's embarrassingly parallel.
We didn't pay her for the advertising.
Yeah, it's true. It's true. I'm just a fan. And yeah, so it's embarrassingly parallel, this setup, because basically I'm running the exact same code with different input variables, different parameters. I would like to petition to change the name, though. I think it should be conveniently parallel or something else. It's just a little degrading. So basically, I feel like you all know this, but the setup is we have jobs, and we feed those to the Niagara supercomputer, which then splits them across a certain amount of processors or cores to run each job. And then if one job takes longer, it will still keep running, but the other cores will accept new jobs, and it basically optimizes the running time. So what was really nice is that I had this coded in MATLAB. And what's nice about Niagara is that it interfaces with a lot of different coding languages. So I didn't have to learn how to code in a more primal language. I simply changed MATLAB into Octave, which is a free version that's pretty much one-to-one compatible with MATLAB. And then there's an interface on Niagara that links up perfectly with Octave. So to automate the process of using different parameter combinations, I simply edited the simulation in Octave to accept command line arguments. So this is my Octave code, and I feed in command line arguments. So that'll be the different parameter combinations that I want to try. And basically it says that if there are command line arguments that are of a certain length, then it's going to update my parameter values to take in those numbers. And it continues on and says else use default, just so it would run on default if I didn't give it command lines. So then each full job is the command line instructions for Niagara. So in whatever interface you plan to use. So here my command is Octave. So it's, if anyone's used R script, it's the same idea. It's just telling Niagara you're about to run an Octave script. And then you tell it which file you want it to run in Octave. And then these are my command line arguments that get fed in and become my parameter estimation tests. Okay, so then we have our jobs in a list. And I would have hundreds of these in one. And creating these lists can also be automated in your favorite coding language. And then what we need to do is feed that job list to Niagara. So this is my batch submission that I actually submit under S batch. And this would be the job. So I think this is just basically telling it to read in bash language, and then we have some commands on running time, how many nodes I want, how to split the tasks, just a job ID, and then just email me everything about this. And so you first need to tell Niagara to set up the workspace that you're going to want it to run those jobs in. So again, you're telling it to load a module, and here you can just find these online. There's a list of if you want to run an R, this is what you have to set up the module for. So here we have Octave, and then the other key component is to tell it to run this new parallel package. And that's what actually allows it to split those jobs up at the same time and optimize the running. And so these are just, the second line is just commands for the parallel computer. So just compressing it, telling it how many jobs, and then I give it the job list. So you'll notice that this title of my list matches this title, and so it's simply feeding every single job I have into this process.
How long do those take to run each? About?
It depends, but some of them were 10 minutes and some of them were 20. Yeah. And we were running it for about 600 years because we were trying to actually get the disease to reach an equilibrium. And that was our metric that we were comparing for prevalence. So yeah, on my computer, some of them would take all night. So it was definitely worth it to have it set up like this. And then what did I get out of that? So this doesn't look like much, but it actually says a lot. What it would do is each job would spit out a CSV file that would give me the prevalence of each age class at equilibrium. And so what I was able to do is, so this is simply for model B. On the y-axis, we have the percent of diet that's coming from cannibalism. So we explored a range of up to 2% of their diet coming from cannibalism, which is a lot. And then from conspecific scavenging, same idea. So we were actually able to, within each figure, we explore that space, and then each figure, there's 3 other parameters in that model. So as we go across each figure, those other 3 parameters are changing. And so this was, I think there was over 10,000 model runs that went into creating these contour figures. And yes, they're all black, but what that tells us is that across no parameter space that we explored does this model structure actually allow you to get a prevalence at equilibrium in the population, which is actually pretty awesome because it means that cannibalism, those intraspecific mechanisms, aren't doing it. So,
but does it mean that they don't eat themselves, or that's not the way of transmission?
Not the way of transmission. Yeah, it doesn't, because all of those explored different levels. They were eating each other. And so if you actually look at the individual data, pretty much when you get high cannibalism rates, population growth rates in bears is always teetering around 1. So if they're eating themselves, most of the time the population actually goes to extinction over hundreds of years. So yeah, it's not a very good method of infection.
Is it the last resort that they would resort to cannibalism? Or?
So a lot of the cannibalism events, they're mostly adult males killing cubs. And again, the cubs don't harbor the infection. So these events don't make a lot of sense as a mechanism of transmission. But under food stress, it's hard to say. You know, some people will say they are cannibalizing more. Some people say that there's just not a lot of information.
Sorry, I just had a question.
Yeah.
You have 2 models.
Yeah.
The previous graph, is it assuming let the only way they can catch these worms be the intraspecific?
Yes.
Then what's the parameters that make the model spit out what's actually observed? Is that what it's going on? Because then I was wondering, like, do you have a model where they can catch a worm both intra and inter and then try to distinguish how much from each?
Yeah, so this one is just interspecific. And then the previous results were considering only intra-specific.
So I know you find that just the intra-specific, there's no parameter
Yes.
values that make the model work.
Yes.
But what if you could combine the two?
Yeah.
and
So-
then there's-
You could, and that is something we considered doing. But when we ran Model A, it's massive what they can get from seals alone. So this is our prevalence in the total population. And again, what we have on the y-axis is from carrion, so transmission from carrion. And then on the x, we have seal transmission. And then across, these are just different probabilities of infection given contact. So this is a very low. Basically, even if you're coming in contact with a seal, you're unable to get it. So we don't get the 60%. Oh, the actual prevalence in the population is 60%. So we don't see it here, but we see that we're already able to get, around 30%, even with that very low probability of infection given contact. And when we move up to a moderate probability of infection given contact, seal transmission is easily able to give you the 60%. And also you can see that moving vertically, moving along the y-axis, doesn't change anything. So the carrion transmission is negligible. The only real changes in the system come when you play with the seal transmission parameters. And yes, we could add them together, but we just didn't feel the need at this point in time, simply because the seals were able to do it so easily. And the reason that seals were dismissed before is because the prevalence in seal populations is 0.6%. So without actually running these models, it wasn't intuitive that this low prevalence was going to be able to create such a high prevalence. But then when you think about it logically, once they get infected, they're infected for life because they have encysted larvae. So they're always testing positive with the seroprevalence surveys. So it's almost like a buildup, like a trophic buildup. Bears are living for 20 years. They just have to encounter one infected seal. So yeah, it was kind of a nice parsimony exercise. Instead of bears having to be cannibals, they just have to eat their normal food source.
Is there a trichinosis problem among Inuit?
It's not too bad to nowadays, but it used
Because
to be.
they consume raw
Yes.
seal.
They boil it a lot now.
Okay.
Yeah, and I think that was the problem.
That used to happen a lot with pork.
Yes.
Certainly in Latin America.
Yes.
I grew up in Mexico. That
So-
was always a problem.
It's the same. This is T. nativa and it's T. spiralis in the pork, and it's still a problem in some. Yeah. So yeah, we can learn that seal seems to be the most probable method of transmission, and it also lets you think about future dynamics of this disease, because we know polar bears are getting a little food stressed. They're switching their eating habits, and polar bears would preferentially eat fat off seals, but under food stress, they're eating a lot more of the muscle tissue, and we know that the cysts are in the muscle tissue. So that probability of infection given contact, some of these parameters might change given that bears are going to be eating more striated muscle tissue. So just very simple modeling exercise that we needed no extra data for, and we can tease apart a lot of what's going on in that parasite system. Okay, so the second case study, the second time I used SciNet. So my major focus of my PhD is determining how polar bears assimilate and use energy for biological processes. So biological processes can be reproduction, growth, movement, which activity, or somatic maintenance. So yeah.
Sorry, so I find this like super interesting, but big picture, let's say you're applying for the grant, like how do you say like this is actually useful? Because you said that people don't really eat polar bears anymore. Polar bears don't seem to be affected by these things. So like, what's the
Big
why
picture?
do we care about? Like I personally find it super.
Yeah.
interesting.
No, fair. because we're writing it up and we're having that problem too. Because it is from a nuance, from the specific position, it's cool. What we find neat about it is that this is a debate that has been in the literature since 1930, and it's basically never been tested. It's just been, here's some data, when we look at it, this is what we think. And so by formalizing hypotheses in a mathematical framework, we're actually able to interrogate that data.
Do you have an idea about like what's the best disease that these lessons you learned from here could apply to, where it actually affects like things we sort of care more about, like people getting infected with like other kinds of worms? Maybe you can take this model and apply it to these other things. Do you have like any idea about?
So, yeah, to me, it's more the method than the actual. I mean, it's neat that we can kind of end the debate, but I think the framework is what's neat, because if you have information on those other worms, you can use this process of translating hypotheses into mathematical equations and then looking at the outputs to try and solve the mysteries in those other diseases. So I think that's what I find more cool, is the process, not the actual information.
So you thought you were a biologist, now you're a data scientist.
Working on it. Working on it.
The fact that the bears don't die of it probably gives you more data to work with. So that's
Yes.
a good thing.
Yeah, and actually, after we've kind of talked about this one, we were able to reach out to the Greenland population, and they actually have kept tissue samples and everything. So we might be able to get burden, the worm burden, and then do a little more modeling with it. But I think it's the process. That's what's really cool to me, is that you don't need to necessarily go get more data to answer some questions. You can use what you have in a different manner. So yeah, that's what I'd say. All right, second one, try and blow through this a little bit. I've taken longer than I thought I would. So we talked a bit about climate change in terms of what it means for bears. So here's a bit of a breakdown. So we have climate change. It's leading to reduced sea ice. This means that bears have less quality of ice to hunt on, so they're getting less food. And they also have a longer fasting period because the seasons of freeze up and break up are changing. This leads to reduced energy in the individual, which can lead directly to higher mortality in those individuals, or increased cub mortality if females can't look after the dependent cubs. And it also means that females are lighter and they have fewer and lighter cubs, which are actually less likely to survive, so more increased cub mortality. So the increased mortality and the decreased reproduction can lead to population level declines. And the link between these changes in the environment and the population is the energy at the individual level. So by understanding how bears assimilate and use energy, we can understand how their survival and reproduction will respond under never before seen energy conditions. And there's a great framework for this, dynamic energy budget modeling. It's a proven framework for linking energy across levels of biological organization. And it's based on the laws of energy and mass conservation. It's used a lot in invertebrates and fish systems, but it's a little harder to do for complex vertebrates. So what I have done is created a polar bear dynamic energy budget model. So specifically for polar bears, I have 3 state variables that I track in DEB models. And for our polar bears, that is structural mass. So that's the components that you can't break down once you build, organs, bones. And then we track energy stores, and that's the fat that you can use at a later date. You can build up and use for energy sources later on. And then reproductive output, so the number of cubs in this case. And then if we do have dependent cubs, we also have to track the cubs' structural mass and energy stores, because the mom and the cubs will affect each other's energetics. So energy fluxes control how these state variables change within an individual over time. And they can be positive or negative. So I consider two positive sources of energy, incoming energy, and that's resource from seals and also milk energy for the cubs. So that's our positive energy sources. And then our energy drains are activity, somatic maintenance, structural growth, and reproduction. And so activity and somatic maintenance are mandatory costs that are prioritized first. You can't live unless you're maintaining your cells. And then structural growth and reproduction are modified based on the individual's energy stores because they don't necessarily have to reproduce. That'll depend on their current energy level. So when I put it all together, I get a model schematic. We have, so this is one bear. You're just tracking at the individual level. So we have incoming resources. Immediately energy is taken out and goes to pay for activity or somatic maintenance. If it's not enough to cover, you have a flux that comes out of energy stores to cover the deficit. If we have a surplus of incoming energy, it can either go to energy stores or to structural growth. And then if you have a certain energy level over a threshold, you can commit that energy to either creating cubs or just giving milk to those cubs. So those are actually, every one of those is a mathematical equation. And they depend on constant value parameters and state variables and individual age, which both vary over time and by individual. And the goal for me is to find out what the constant value parameters are so that we can just plug in a bear of any age or sex and give it a certain amount of energy and see what it does. So when you put these all together, you end up with a set of paired ordinary differential equations, one that tracks the change in energy stores in an individual, and another that tracks the change in structural mass over time. And these are paired because there's a connection that occurs where energy is either split to energy stores or to structural growth. So these affect each other. And then any dependent cubs are also tracked in a similar set of paired differential equations. And so with these equations, I can use repeated measures on individual polar bears, so longitudinal data, to estimate parameters for my model with classical Bayesian estimation techniques, which is great, but that doesn't deal with a change in the number of cubs. This throws a wrench into classical estimation techniques. And we can talk about it later, but it's a problem. So basically I had to either write my own code for estimations, which I cannot do, or in the absence of that, a parameter estimation technique called GLUE was suggested by one of my committee members. So what GLUE is a generalized likelihood uncertainty estimation. And this rejects the idea of one single optimal set of parameters and instead explores the possibility of equally valid sets by running the model with different combinations of parameters. Sound embarrassingly parallel? So you did see the equations. We have lots of parameters. So I end up with about 5 parameters to test after Bayesian. And I draw on experimental data from model systems or controlled environments to establish minimum and maximum values for each of these target parameters. And then I draw values for each parameter from within those ranges to create the sets to test. So to produce unbiased sets of parameter combinations, I use Latin hypercube sampling. And it samples parameter values from a multi-dimensional distribution and gives us confidence that we have correctly sampled the parameter space. Okay. So state variable values that are simulated from each parameter set are then compared to a behavioral space for each state variable. So here I show the behavioral space for structural mass of female and male bears, which indicates observed maximum and minimum values for that state variable structural mass across a bear's lifetime. So then when I look at the output from my different from my model runs with the different parameter sets, I can look at if the simulated values for state variables fall within that behavioral space or without. And that allows me to reject sets of parameters or keep sets of parameters. So help zero in on the correct parameters for each. So yeah, that's why we're going to use SciNet again. So same idea. We can make a job list for each bear that tests each parameter combination for each individual. And then I've done this one in R Studio, and it has a direct interface with Niagara, so there's no changes necessary. Again, I just needed to automate the process so it could accept different command line arguments. So it's just a slightly different command, but exact same idea. If they're there, those new values are substituted in for the parameter values. Okay. And then the Latin hypercube sampling, it's also automated. So all I need to do, there's a package already in R, and you pick the number of points you want to explore between the minimum and maximum for each. And then you just give your minimum and maximum for each parameter. And we end up with a table. It pops out a table, and each row is a parameter combination set. Okay. So same idea. This is our command line, our commands for Niagara. So here we're going to tell it to run our script, give it the file, and then our command line arguments for parameters. Okay. And again, similar idea. We have a job list. We feed it to Niagara using our sbatch command. So I'm just kind of burning through this because you guys know there's just a slight difference here because we're telling it to work in R instead of Octave. Okay. So what I get out of each run is a CSV file. And this is for one bear. And what it does is it gives me a CSV file for every year that it ran. So if the bear lived to 15 years, I have 15 CSV files in a folder. It gives me the time point, the energy scores of the bear, the mass, the structural mass of the bear, her age, any dependent cubs, and then any dependent cubs statistics. And so what we can do is, for example, this is one model output from a bear, and this is her structural mass, so it's an adult that never changed. But you just compare it to your behavioral space and make sure that it fits within there. And so I would keep that set of parameters based on structural mass. I'd do the same for the energy stores and make sure that it fit all parameter or all state variable space. So I guess what I'm trying to say is that SciNet wasn't as scary as I thought it was going to be, and I didn't know it existed until 2 years ago. So it's a relatively simple step from coding in common languages to implementing these programs in the supercomputer. And you can use the interface of your choice, so you're still coding in your favorite language. With minor tweaks, we can save a lot of time doing sensitivity analyses, both global, so across all parameters, and local, looking at one. We can help estimate parameters, like with my glue example. And it's great for model comparisons. You can run the same code, or you can run the same parameters with different structures, different mechanisms, see what makes sense. And basically, ecology can really benefit from this, as there's a lot of uncertainty. And it can help us to work better with field biologists. And we have this interface where we can use the model to direct fieldwork and get things back from fieldwork, and they can direct us on what they think is important. We can explore it in model simulations. So saving time and money.
Wow.
Yeah, sorry. I didn't think it was going to take that long.
We gotta use you for our marketing.
Yeah. Yeah. So. All right. Thanks for listening, guys. And yeah, I really enjoyed working with coding now, so it's been fun.
And the question is, would you have been able to do it on your own?
What do you mean?
Or only only using the help of Eric and Marcelo and those nice guys.
So, yeah, that's another benefit of SciEx.